Good morning and good afternoon, ladies and gentlemen. A warm welcome to the latest installment of the CDX Engage webinar series, hosted by Diagnostics, entitled A Look at the Future of Blood-Based Companion Diagnostics, Rapid Development of PMA-Ready Clinical Assays. My name is Ben Carrington. I'm from the CDX Engage team here at Hanson Wade and will be chairing our session today. This webinar will be 1 hour long and consist of 2 presentations followed by a Q&A session. Please submit all questions via the questions tab at the bottom of your webinar dashboard. We will endeavour to get through as many questions as possible in the allotted time. All unaddressed questions will be passed on to the speakers, so if we do run out of time, they can follow up with answers directly. To deliver these 2 fantastic presentations, it gives me great pleasure to introduce our 3 expert speakers. Firstly, Gary Pestano, Vice President of Development and Operations at Diagnostics. Gary is the New York State CLEP Laboratory Director for Diagnostics' CLIA, CAP, CLEP, and ISO certified laboratory. He has vast experience in high complexity diagnostic tests, including molecular and proteomic testing. Secondly, Viresh Patel, Global Marketing Director of Digital Biology at Bio-Rad. Viresh leads a marketing group responsible for diagnostic product development and commercialization of Bio-Rad's droplet digital PCR portfolio. He has held multiple roles in marketing with Bio-Rad and MJ Research and has managed a diverse group of genomic and gene expression product portfolios. And finally, Dawn Shelton, R&D Manager for IVD products at Bio-Rad. After completing her PhD at the University of Utah and working in a clinical cancer research lab at the University of Iowa, Dawn moved to Bio-Rad to help develop the droplet digital PCR platform. She is now an R&D manager for IVD products at the Digital Biology Center, focusing on developing applications and collaborations for ddPCR in the cancer field. Gary, Viresh, Dawn, a warm welcome to you. To quickly run through today's agenda, we will start with Gary talking to assay development. Viresh will then take us through partnership models before Dawn presents a new assay and MSI case study. Gary will then wrap up with a quick summary before we have our Q&A session. Now, without further ado, we will begin today's session and hand over to Gary.
So thank you, Ben. Thank you, Ben, for that introduction, and thank you all for attending today. On behalf of Diagnostics and Bio-Rad, I'm happy to have this opportunity to update you on some of the work that we're doing here at Diagnostics in terms of ddPCR and our partnership with Bio-Rad. First off, outside of having disclosures as an employee and a shareholder in Diagnostics, I will not be discussing any off-label use or investigational use in this presentation. So a brief introduction to Diagnostics, and for today's focus on technology and technology partnership, including laboratory developed tests, I'd like to introduce Diagnostics and how we look at things from the perspective of being an LDT-focused diagnostic company. Really, we address multi-omic technology capabilities and look at developing custom biomarkers. So our focus primarily is in blood, not so much in tissue right now. And we use a variety of technologies. Today, of course, we're talking about ddPCR. NGS is another application for another day, but also quite a bit of effort in the field of mass spectrometry. The key point being to partner with innovative technology providers, and you'll hear me say this quite a few times, who have high-quality approaches to development of their technologies and platforms, where we can bring our expertise in test development to bear. So this really highlights or leads into challenges in the commercialization of lab-developed tests. So in our experience, one really needs, in order to be successful, competencies in all of what's shown here: discovery, development, having a strong quality and regulatory infrastructure, of course, supported by a field team, med affairs, a sales team, for example, as well, if it's a part of the strategy, and an underpinning in supporting test development using clinical studies, clinical trials, and reimbursement, which really helps the situation in terms of being commercial. So for today, we're going to focus on the development aspect heavily, not so much many of the other parts of the infrastructure, but just to highlight that our laboratory, which is a centralized laboratory based here in Boulder, Colorado, is governed by CAP CLIA, the quality system by ISO 13485, and all of the tests that I'll be talking about today are also approved by New York State CLEP. So our on-market tests are known as the Biodesix Lung Reflex, and contained within that are two portfolio of tests, one in mass spectrometry, a high-value prognostic test for outcomes in the VeriStrat portfolio, and then in GeneStrat, where we'll be looking at the ddPCR test for DNA and RNA. So why have we chosen to partner with technology providers? Well, a key point is that we found here at Biodesix, it really enhances the quality that can come to product development and eventually onto the commercial market. Also, of course, it improves critically the time to getting a test ready for either a pharma clinical study, a research study with key opinion leaders, or in the commercial testing marketplace, all places in which we use our tests. So getting a little bit more into what specifically we have done with Bio-Rad is utilizing the ddPCR platform and content provided by Bio-Rad is really have an ability to access new and improved content. We, in this partnership, both parties, of course, provide expert assistance in assay design, in silico analysis development, and eventually testing with clinically relevant controls and specimens. As things transition to Biodesix, you'll see here in terms of assays, under our quality management system, we really follow the design control process in delivering compliant assay development. And then together, we can access globally commercial channels. So the bottom line is we engage with quality partners who have innovative technologies, and overriding all of that, though, a very, very strong interest in the clinical diagnostic testing market. So a little bit more about how this looks in the graphical format. We can see very broad strokes. Of course, there are overlapping areas. Bio-Rad has a focus in biomarker discovery, selection, feasibility, and some other aspects of this depending on what is being developed. We'll talk a little bit today, or the Bio-Rad colleagues will talk a little bit today about the QX200 and its progress with FDA. But at Biodesix, our focus is really on assays that go onto that platform. And we generally start in late stages of feasibility in our collaboration, but definitely in verification, validation, and in our case, providing commercialized laboratory developed tests here in the United States. So what have we been doing in our experience with looking at circulating DNA and RNA? Again, the focus for the ddPCR talk today. This is summarized in a recent independent review conducted by an author for the Medical Laboratory Observer. So this publication was from June of 2017 and conducted a survey of up to 6 different companies providing molecular diagnostic tests for hotspot mutations, fusions, etc., both tissue and in blood. And I want to draw your attention to the company shown as Company 1. I can jump ahead and tell you that's really Biodesix. If you look at the reference that links to that, there is a key publication that my colleagues and I had reported development of our circulating DNA and RNA tests in blood. But certain features are highlighted here. So the platform, of course, is ddPCR, sensitivity and specificity, which also are listed in the publication, are shown here in the high 90s, specificity at 100% for the various tests. But a key point is for blood testing, we can turn around results to physicians from time of receipt of samples in 72 hours or less. So where did this all start? First, our focus was on typical hotspot mutations, if you will, as blood-based testing was coming into its own. And our focus here with Bio-Rad was on developing for the clinical market, pharma as well as the commercial market in the United States. Tests for hotspot variants in EGFR shown here, KRAS and BRAF. So of course, Bio-Rad's approach here is to provide the technologies and the platform, while Biodesix provided verification, validation of the test for clinical use. As it says here in the footnote, all of the tests in GeneStrat shown in this table are also New York State method validation approved. A key thing for me to highlight, and you'll see this in a few slides as we hit on the references, at Biodesix we have a very, very strong publication strategy. So while I may not be highlighting all of the data aspects around the validation of the tests shown here, I do want to point out the sources for the actual data, and we can follow up with any technical explanation needed around how these tests were actually validated for the clinical use. In an advance, Bio-Rad has developed an expert design tool for more high complexity assays, and so our next foray into the blood-based testing market was to develop RNA detection assays. So shown here is a table of our, again, approved tests for looking at fusions in ALK, ROS1, and RET. So the gene targets are shown in the middle of the table, and then the fusion partners, numbers of transcripts that we are capable of detecting are shown in bold. So for ALK, in a single tube assay, we can detect the 3 most prevalent variants. ROS and RET, both we have up to 8 different variants, and I'll show you the coverage in a few minutes that can be detected in these single tube assays. So again, using the Bio-Rad design portal, these assays can be developed very, very rapidly. On this slide, I also want to emphasize the publication that went along with the RNA assays, and we think this is something that's really, really educational and at the same time cool. So I encourage any in the audience to look at the publication we had earlier this year on RNA detection in blood using ddPCR that was in the Journal of Visualized Experimentation. If you're not so familiar with this journal, there's a video that accompanies and shows in this case exactly how to conduct RNA testing in blood. So to get to some data for EML4-ALK and its analytic and clinical validation that was conducted here at Biodesics, you can see 2D plots. My colleagues at Bio-Rad will go more into how ddPCR is conducted and details of resulting, but just in a snapshot, so shown on the left, the lower panel really highlights on the y-axis EML4-ALK fusion positive variants, and so those are the blue dots or droplets, and on the x-axis we look at a control gene, GUS-B, which is a required quality control to say we have enough RNA in circulation to be detected. The darker dots are negative, if you will, or empty droplets. So in terms of analytic validation, the tests that we've developed, whether it's DNA or RNA, have a sample ship stability. We use the STREC DNA and RNA tube in the development of these assays. So sample ship stability is up to 7 days in transit. Using services such as FedEx, we generally get samples more than 99% into Biodesics and prospective collection within 24 hours. So 7 days is not really a risk, but we've certainly validated the samples in transit up to that time. In terms of limit of detection, it's variable, but for most of the assays, it's 0.2% or below. Again, I can point you to those technical details within the manuscripts. Importantly, clinical validation, again, is a necessary part of doing the development of these tests, and we can see using reference samples, in this case from tissue, for the validation of EML4-ALK, we achieve specificity of 100%, which is not unexpected for PCR-based assays, sensitivity and concordance at 85 and 92% respectively. In another case study of the high complexity multiplexed assays, and I just want to show an example of ROS and then followed by RET. And again, the key point here is not only to focus on the 2D plot, which shows, for example, variants positive for ROS1, but also the coverage here, which is up to 88% of the known prevalent variants for this gene fusion. And in the next example, even more impressively, where we've achieved 99% or higher prevalence coverage with the variants in this multiplex assay. In the final clinical example of developing these high complexity assays, I wanted to highlight our EGFR del19 variant assay, which detects up to 15 variants within EGFR del19. This was reported by an independent investigation conducted by a lead investigator from AstraZeneca at the time, Ken Tress, and reported at ASCO last year. So this shows potential clinical utility for ddPCR in monitoring efficacy, so looking at the blood and circulating free DNA and its prevalence after treatment. This happens to be with Tagrisso. And so what we're looking at in the Kaplan-Meier curves are performance-free survival at 3 weeks and performance-free survival at 6 weeks. And what you can see in the blue lines, when there's still detectable mutations at 3 and 6 weeks, those patients perform less well than the ones who had achieved clearance of cfDNA and as early as 3 weeks. The Del19 variant assay in this format is also approved by New York State. That concludes my presentation of what Biodesix has been up to in the space in ddPCR. We, of course, continue to develop more assays, and at this time I'd like to turn it over to Bio-Rad. I'll return at the end for an overview of where the two companies are going next. So thank you for your time.
Okay, thank you, Gary, for that introduction and presentation, and thank you to all who are attending today. My name is Varesh Patel, and during this portion of the presentation, I will start by reviewing a few partnership models with Bio-Rad. I'll then dive into a little bit of background on our droplet digital PCR technology, a brief review of a portfolio of our products, before handing off to my colleague, Don Shelton, who will describe a new assay currently under development here at Bio-Rad. At Bio-Rad, we see a variety of models that diagnostic test manufacturers can utilize to develop and make available diagnostic or companion diagnostic tests to determine drug therapy selection. Bio-Rad's deep expertise in IVD product development and complex regulatory landscapes enables us the flexibility to partner in multiple ways. The first option that Bio-Rad offers involves a distributed kit model where Bio-Rad takes the diagnostic all the way from ideation and biomarker discovery through to kit launch. A second model that we have just heard from Biodesix that we'll explore in more depth today is one where Bio-Rad is partnered with Biodesix throughout biomarker discovery, development, and test commercialization. We just heard from Dr. Pestano how Bio-Rad and Biodesix have successfully partnered to expand the on-market GeneStrat test menu. By employing this CLIA lab model, it allows for the rapid development and deployment of diagnostic tests in an already regulated and approved environment using Biodesix's CLIA-certified laboratory to both run and monitor test performance. We will discuss in depth how this model aligns well with the phases of therapeutic development in just a few minutes. But first, to get everyone familiarized with droplet digital PCR, I want to spend a few minutes providing some background of our technology, showing its adoption across a wide range of clinical applications, and briefly reviewing our broad portfolio of ddPCR offerings. So here is showing the evolution of PCR technologies, starting 35 years ago with the advent of PCR, which was qualitative in nature. Then about 10 years later, qPCR was developed and introduced in an effort to make PCR more quantitative. Now, these two technologies share one common principle, that is a principle of measuring the sample in bulk or making one measurement on a biological sample. In 2011, Bio-Rad introduced droplet digital PCR, which changed the paradigm from indirect or relative quantification to a direct or absolute quantification. And this is achieved by changing the measurement paradigm from a single measurement into many thousands of discrete measurements. And Bio-Rad utilizes droplet partitioning technology to achieve this. We call this the power of partitioning. And what I'm showing here is a graphical depiction of a typical nucleic acid detection schema where, in the case of cancer mutation detection, you may be interested in detecting a small number of mutant molecules in the background of several thousand-fold excess wild-type molecules. So on the left is shown a mutant abundance graphic of 0.1%, or 40 mutant molecules in the background of 40,000 wild-type molecules. PCR and real-time PCR would be challenged with this to detect the mutant molecules in this complex background, primarily due to competition of primers and probes for the relevant and appropriate targets. Now, in droplet digital PCR, this sample is partitioned into thousands of droplets. Depicted on the right is what many of these droplets would look like. In addition to droplets that contain no molecules of interest, a vast majority of the droplets would contain mutant wild-type molecules only, and a subset of these droplets would also contain mutant molecules. And what you'll notice that in each of these droplets containing a mutant molecule, the relative abundance of mutant molecule to wild-type molecule is dramatically enriched to about 1 in 3. This presents no problem for PCR to detect in this case. And so in this example, droplet digital PCR can dramatically increase the sensitivity of nucleic acid detection by increasing the local abundance of rare targets. And furthermore, droplet digital PCR increases the precision of nucleic acid quantification by making thousands of discrete measurements. The workflow for droplet digital PCR is very straightforward. Users would prepare a reaction as they would any real-time PCR or PCR reaction. However, prior to amplification, this reaction is partitioned into droplets using one of two instruments that Bio-Rad provides, a manual droplet generator, or, as is shown in the picture here, the automated droplet generator. Following partitioning, the reactions are then amplified on a standard thermal cycler to endpoint. And following amplification, these droplets are then put into a droplet reader for individual analysis. This droplet reader is akin to a flow cytometer-like instrument, which interrogates individual droplets in each sample and measures the fluorescence, counting those droplets, and then providing a nucleic acid quantification readout. The key advantages of droplet digital PCR, the first advantage we highlight here is precision. This technology has the ability to discriminate 10% differences in nucleic acid concentrations. This translates into high reproducibility, low variance on replicates, and has been demonstrated to be shown in multiple interlab studies as a highly reproducible technology and test or assay, regardless of operator or regardless of site. I mentioned previously that droplet digital PCR provides absolute quantification. This means that no standard curves are required, as is often the case in real-time PCR. There are also ways and strategies to multiplex droplet digital PCR assays so that samples can be maximized. I just showed the example where sensitivity is enhanced, especially in the case of detecting rare targets in complex backgrounds. And finally, because droplet digital PCR is an endpoint analysis, it is often rendered resistant or resistant to PCR inhibitors that are commonly present in nucleic acid samples. The clinical landscape of droplet digital PCR applications is broad. It has been applied in both clinical research and in clinical testing across various areas that are shown here. And for the purposes of this presentation, we'll certainly focus on the applications of droplet digital PCR in oncology, and as Gary has mentioned, in liquid biopsy. The technology has shown broad adoption and rapid adoption since its introduction. To date, we have tabulated over 2,800 research publications citing the use of droplet digital PCR in applications ranging from basic research, applied research, all the way through to clinical research. And it's no surprise here, given the advantages of droplet digital PCR, that over 900 of these publications are in clinical research journals. And what's even more astonishing is that 750, or about a quarter of these publications, are in the area of liquid biopsy, and this spans beyond the areas of cancer into transplant monitoring, as well as diabetes. Now, in addition to a broad range of research use only portfolio and products, this slide shows Bio-Rad's current portfolio of diagnostic products. On the left, I'm showing the availability of a QXDX droplet digital PCR systems, universal reagent kits, as well as our first digital PCR-based diagnostic test, the QXDX PCR-able test. These are all available outside the U.S. as CE-marked products. And on the right, I'm showing the soon-to-be available QXDX AutoDG system and QXDX PCR-able %IS test, which is currently under review at the FDA. In addition to the platforms and tests that I've just described, Bio-Rad offers a broad range of research use only assays, ranging from wet lab tested and wet lab validated mutation detection assays, copy number determination assays, as well as gene expression assays. We also have available various multiplex kits for some of the more commonly studied cancer variants. And finally, we offer assay on demand services for novel biomarkers that may be discovered in your laboratories to have assays designed for droplet digital PCR. As Gary described, the expert design portfolio, part of which was used to help expand the GeneStrat menu. Currently, Bio-Rad offers 41 assays. Now, these are ranging in various stages of complexity, but are typically more high complexity assays, highly multiplexed assays, and these are also available for downstream studies. If we don't have an assay available for you, Bio-Rad also offers custom assay design services, and that is something that certainly we can engage in as we go forward. So now let's dive into a bit more detail of Bio-Rad's product development process and how it lines up with therapeutic development. We employ a rigorous phase gate approach to diagnostics development, starting with biomarker discovery, selection, often through strategic collaborations. Once a biomarker is identified for either diagnostic or companion diagnostic, feasibility studies are conducted to develop a prototype assay that can be distributed as an RUO labeled product to enable preclinical research studies. Once the drug passes preclinical stage gates, Bio-Rad can develop and verify the biomarker test in preparation for phase 1 and phase 2 clinical trials. Finally, as phase 3 trials progress, the Bio-Rad companion diagnostic will be validated in conjunction with the drug hypothesis and submitted to the FDA for regulatory approval and subsequent commercialization. All of this is done under strict design control, considerations for 21 CFR Part 11 compliance, and ISO 13485 regulations to ensure consistent quality. FDA guidance issued years ago directs pharmaceutical companies to have diagnostic tests available to ensure proper patient selection. This model has demonstrated not only a direct benefit to patients and payers, but can also significantly accelerate the path to drug approval and commercialization. The development process that we have described allows Bio-Rad and Diagnostics to work together to achieve rapid diagnostic commercialization that is aligned with the therapeutic development process. In phase 1, Bio-Rad, together with the pharma company, is responsible for biomarker discovery and creation of the initial assay, including identification of appropriate controls. As successful drug development continues, Bio-Rad's IVD design control process is utilized in phase 2 to complete assay development, including instrument integration of our QXDX system and analysis software. All design control elements, including analytical sensitivity and specificity studies, reproducibility, and control selection, are executed prior to RUO prototype verification. Once analytical performance is established, development of the IUO test ensues with the completion of design verification, software and process validation, finalization of the design master record and design history files. This allows for transfer of the IUO test to Biodesix in order to perform clinical trials and complete clinical validation of the diagnostic test. Upon completion of the clinical validation in phase 4, Biodesix and Bio-Rad will together submit all required information to the FDA. Our QXDX digital PCR system is expected to be cleared in late 2018 and should not require significant additional documentation for the approval of a jointly developed companion test that Biodesix may submit. Once FDA approved, Biodesix can then commercialize this companion diagnostic and perform all required post-launch surveillance in their CLIA laboratory. Now that we've described our development process, we'd like to switch gears and tell you about a new test that we are currently working on. As shown here, biomarker selection and assay creation can originate from a variety of sources, including collaborations with key opinion leaders within academic research groups, pharmaceutical companies, or clinical research laboratories, all of whom have the ability to create their own assay or have Bio-Rad create one on their behalf. Once a biomarker or biomarkers have been selected, feasibility studies are initiated to optimize assay performance before moving downstream into verification, validation, and commercialization. And with that, I will hand it off to Dawn Shelton, R&D Manager in our IVD program, who will describe a current project focused on assay development for biomarkers used in immunotherapy selection.
Thank you for the intro, Paresh, and the thorough explanation of our process. I'm going to introduce a little bit about microsatellites. We're developing an assay for microsatellite instability. This is an example of an assay we are currently developing. Microsatellites are repetitive sequences scattered throughout the genome. They consist of mono, di, tri, and tetranucleotide repeats. The mutation rate across these regions is higher than the rest of the genome due to polymerase slippage on these repeat elements. Extra repeats can be either inserted or deleted. With functional mismatch repair genes like MLH and MSH, these errors are generally corrected. But when one or more of these genes are mutated or silenced, microsatellite instability occurs and the mutations accumulate at a higher than normal rate. These mutations can then lead to the production of neoantigens, which can be targets of the immune system and immunotherapy. Current methods for diagnosing MSI include both cause testing and effect testing. Cause testing evaluates the functional status of MMR proteins by immunohistochemistry, shown here. These are performed on tissue slides from tumor, but they can miss cases where the protein is produced but not functional. Effects testing evaluates a standard set of microsatellites and looks for changes in microsatellite size by capillary electrophoresis. It's most commonly used with a panel of 5 to 7 markers. These evaluate the target length by PCR and capillary electrophoresis. 2 out of 5 of the markers, greater than 2 or more of the 5 markers positive, or 30% more of the markers would be diagnosed as MSI high. 1 out of 5 markers positive, or 0 to 30% of the markers being positive, would count it as MSI low. And 0 out of 5 markers would mean that this is considered microsatellite stable. There are other testing methods that exist, which are currently a little less established, but being evaluated. NGS, which is expensive, has a long turnaround time, and is not as sensitive and is very bioinformatics heavy, and ddPCR, which is low cost, easy to use, has a quick turnaround time, as Gary mentioned, and highly sensitive. Shown here is an example of some of our singleplex MSI assays for 5 different targets. The clusters in blue contain mutant alleles for these targets, shown in the circle, and while the clusters in orange represent the wild type alleles, shown in the orange box. So this is an example of some of the data from those singleplex assays illustrating sensitivity, analytical sensitivity to below 0.1%. As you can see, Both of these panels are close to 0.01%. Another format we're working on is an example is our early prototype multiplex assays for MSI. On the left is a duplex assay with 2 targets in blue and shown in blue and green, and their corresponding wild type alleles in orange. On the left is our triplex assay with 3 targets, again in blue and green, and the wild type alleles in orange. These assays were then tested on some matched tumor and plasma samples. So this is a first pass look at detection of MSI in plasma cell-free DNA with ddPCR. Plasma samples were acquired that had tissue results positive for MSI. Only 0.5 mils of plasma was run on an on-market test for MSI and on the Bio-Rad assay. The on-market test using 0.5 mils of plasma yielded 9 out of 10 results as microsatellite stable, in contrast to the tissue results. The Bio-Rad assay yielded 7 out of 10 as MSI high, 2 out of 10 as MSI low, and 1 as microsatellite stable. 5 samples with stars have a very low signal, likely due to the limited input sample and high levels of contaminated genomic DNA. We measured these samples and found that greater than 50% of the DNA, or cell-free DNA in this case, was actually contaminating genomics. As you can see, even with this low sample volume, more MSI was detected in plasma with ddPCR. These assays look promising, are being further refined and tested for future release. And we'll show you more in the future. Thanks, and back to you, Gary.
Thanks, Dawn and Resh, for that presentation. So for our closing slide, I'd like to summarize what's next for both Biodesics and Bio-Rad. Obviously, extending from what you just saw, we here at Biodesics are very excited about receiving access to, as I'm sure many of you are, this emerging biomarker to begin clinical validation in terms of utility studies. So for MSI, a blood-based PCR assay would be very, very valuable. Reasons, again, being early access to making treatment decisions, or in pharma studies in recruiting patients for ongoing clinical studies. Aside from MSI and hopefully bringing that soon to our clinical testing and physician testing market, we're also very, very excited at extending applications such as the one you saw presented here earlier from the data presented by AstraZeneca at ASCO last year. So looking at things like minimal residual disease monitoring, utility for cfDNA, for example, in treatment patients. We're also looking forward to developing more content, highly multiplex, high content assays, and including phenomena such as overexpression. And I think some of you who've been following some of the press that both Biodesics and Bio-Rad have been putting out, you're familiar with our efforts in PD-L1 and measuring PD-L1, for example, as a marker for immunotherapy using ddPCR. You're probably also familiar with the conduct of the prospective study that supports the development of this assay, and we look forward to updating in the future. As Varesh and Dawn touched on, CDX assay development is important for both companies. So availability of the FDA-approved platform supports both of our interests. Having robust approved platforms or cleared platforms, such as the QXDX ddPCR system, is highly anticipated, I would say, for the entire testing community, and certainly broadens our ability to develop tests, not only for the United States, but beyond. So, for example, the GeneStrat EGFR specimen collection kit is already CE IVD registered. We also are, of course, excited by the development at Bio-Rad as they continue to progress tools and technologies and assays of potential utility into the clinical diagnostic market. So a more high throughput multiplex ddPCR platform and various assays would be extremely, extremely helpful in getting results to physicians even quicker and more cost effectively. As I mentioned, an FDA cleared system would have as well many, many benefits. And as Bio-Rad continues to expand their global access with partnerships in distribution and registration, we look forward as well to being a part of that. Working with Bio-Rad has really allowed us to be flexible, as you saw, everywhere from early discovery, target selection for assays, feasibility, verification, validation, as well as on-market offering, and we look forward to continuing this partnership. So at this point, I'd like to say thank you for your time on behalf of both Biodesics and Bio-Rad, and I'll turn it over to Ben for questions. Thank you.
Thank you, Gary, Varesh, and Dawn, for your fantastic insights. We do have some time for a few questions. If you would like to submit a question, please do so in the question box. First question, Gary, is for yourself. What are the advantages of ddPCR versus NGS?
So thank you, Ben, for that question. This was actually addressed, I think Dawn touched on it a little bit in the context of the MSI assay. We see certain advantages for ddPCR relative to an NGS approach, depending on where the test is really being used. I would say if broad profiling is the question, there are certainly reasons to want to do NGS in high volume setting. But as Dawn touched on, there's a necessity for batching with NGS approaches, typically to maximize the cost of that investment and the reagents. So NGS per sample costs are many times more expensive than a typical ddPCR assay. And importantly, though, for the clinical utility question, again, whether it's in the pharma setting or the physician setting, is the turnaround time. So getting results back in that 48 to 72-hour time frame that allows treatment initiation or recruitment into a clinical study to occur quickly can only benefit all concerned. Sensitivity as well was touched on, and depending on the assay we've seen, ddPCR can really outpace an NGS approach.
Thank you, Gary. Dawn, we have a question for you. You stated that NGS is less sensitive than your MSI assay. Can you please elaborate?
You know, we've seen some work that shows that next-gen sequencing in plasma is pretty sensitive on their own, but in direct head-to-head, our assays right now are preliminary. So we've seen good results, and I won't make the grand statement. We are claiming that we're much more sensitive than electrophoresis, capillary electrophoresis right now.
Thank you, Dawn. And here's a follow-up question for yourself again, Dawn. How many MSI repeats do you analyze in your digital PCR assay?
Currently, we're working on 5 repeats at once, but that is under development, and absolutely what's more important is the method of designing the assay has been optimized, and so we can put in as many as we need.
Great. Varesh, we have a question here for you. How long does the CDX development and approval process take?
Okay, thank you. So that's a pretty broad question. So I'll try to answer that in a bit in some smaller chunks. So in the case where Gary described the Biodesics RNA assays for GeneStrat, in the case where he showed the EMO4-ALK, ROS, and RET fusions, those are a fairly complex set of multiplex assays that Bio-Rad designed. This process took about 2 to 3 months here at Bio-Rad to design, test the assay for some basic performance ahead of then providing that assay to Biodesics for then subsequent downstream verification validation. And so that's just an example of how long it takes Bio-Rad to design an assay. Now, the question about an entire companion diagnostic, I think, is a much broader question that would rely on certainly the therapeutic selection and development process, which would ultimately be longer than the diagnostic development process. But just to give an example there, on our side, it would take a handful of months to be able to design an assay, then to hand over to Biodesics for further downstream validation studies.
Great, thanks. Gary, do you have anything to add to that? We have a follow-up question that links in quite nicely, which also asks about kind of the cost to develop and release a CDX test.
Yeah, so maybe I can sort of finish off where Varesh left first, Ben. So I think the real limitation on many of the opportunities for either creating a laboratory developed test as a standalone or a companion, even as we would pursue from a centralized PMA perspective, is the samples that are available to conduct the verification and clinical validation study. So I would say that impinges on time, and so a partnership where samples can be available, preferably from multiple sources and so on, really helps advance the time. And I would say, as everything goes, time has a relationship to cost, and so I would say the costs are also potentially variable. If samples are ready and qualified and can be used, that is a much better situation. If though a prospective collection needs to be conducted, the costs can increase quite significantly.
Thanks, Gary. A follow-up question here. Can you comment on the clinical utility of an MSI assay with a limit of detection below 0.1%? Are you planning to evaluate concordance between plasma and FFP, for example? Dawn, would you like to answer that one?
Sure. Could you actually repeat the question?
Sure. Can you comment on the clinical utility of an MSI assay with a limit of detection below 0.1%? Are you planning to evaluate concordance between plasma and FFPE, for example?
Oh, yeah. So first off, clinical utility below 0.1% still does need to be established. However, the reason you'd want to be looking in that range is if you're doing MSI detection in blood, the vast majority of the sample is actually going to be wild-type DNA from blood. So, that outweighs by far the percentage amount. And so being sensitive down to 0.1% and below is going to be critical if this is the kind of test you're developing in blood, especially if you intend on moving forward, or moving earlier from rather stage 4 cancer to stage 3, for instance. As to the second part of your question, we will be doing a comparison between FFPE and plasma, definitely.
Great, thank you, Dawn. Gary, have you anything to add on that clinical utility comment at all?
Yeah, I think MSI, of course, is an interesting marker as it was approved by FDA across cancers. We know a complication with this biomarker is that it's clinically relevant at variable frequencies for solid tumors. So, in colorectal cancer, for example, it's more frequently represented, and in something like non-small cell lung cancer, less frequently present. So this again comes to, what samples are available with references from FFPE that we can procure for the blood-based test. Is it relevant at all to be organ specific? I think FDA approval broadened that quite a bit for tests like MSI and TMB. But there are additional questions about utility across cancers and, whether or not the MSI marker set that is appearing in colorectal, for example, is the same in terms of clinical utility with lung cancer. And I think those are the studies that need to happen. So I don't know that it's as simple. Maybe it's as simple as concordance with tissue in analytic validation studies. But I think as we look forward to clinical utility studies in tissue as well as in blood, these questions will have to be considered.
Great, thank you. A follow-up question here. Can the speakers comment on the utility of ddPCR for RNA-based markers from plasma? Any examples? Gary, if you'd like to tackle that first, and then we will pass over to Varesh and then Dawn.
Yeah, so I think, the two publications that we've put into, the two peer-reviewed publications, I should say, that we have in literature speak to the utility in the Journal of Molecular Diagnostics, for example, of retrieving RNA. We use a platelet-enriched plasma fractionation approach that was developed here to retrieve anything and everything that is in circulation regarding RNA. So we harvest anything that's attached to an exosome, that's attached to a platelet, or circulating free. So initially, we had done studies, given interest in exosomes, for example, looking at exosomes only, looking at free circulating RNA, etc., and we found no real advantage to going after a specific compartment. So in terms of, RNA really had to begin with methods of detection to successfully retrieve RNA from blood. So methods of development here was extremely important. And again, we've published those methods both in the Journal of Visualized Experimentation for ROS and RET and for EML ALK variants in the Journal of Molecular Diagnostics. So again, I think there were some pretty basic things that had to be done that had to, nothing really to do with ddPCR. The technology was applicable or is applicable to whether you're looking at RNA and DNA from cells or in circulation. The point is, once we have the template, we can then conduct the assay. What is important for the clinical assay, though, of course, is looking at the quality of the sample. So this is why we monitor a control gene, and we've been doing RNA testing now for more than 2 years in the clinical market space. And in another presentation, at some other point, we can talk about stability of RNA over time and drift in the assay. I can tell you all of these assays are monitored, DNA and RNA, and they're remarkably stable from day to day in our clinical lab trending. So Varesh and Dawn, maybe you have things to add that are specific to these tests, but that's the summary of our clinical experience with RNA.
Yeah, sure. Thanks, Gary. So to extend a bit beyond what you spoke about, Gary, yes, our RNA tests and digital PCR are certainly valuable, and maybe I can touch upon areas outside of oncology. There have been dozens of publications citing the use of digital PCR to detect residual HIV or low levels of HIV and other viruses. RNA viruses. So when it makes sense to employ an RNA test where you're looking for maybe a limited number of RNA markers, digital PCR with the sensitivity that it offers and the relative turnaround time and cost that it offers is an advantage. And I'd also like to mention that our first test that we've made commercially available as CE marked is a BCR-ABL test, which is an RNA test from peripheral blood. And this is, of course, taking advantage of the extreme sensitivity that digital PCR offers and precision and reproducibility that it offers to now enable labs to, independent of operator or site, be able to get the same answer over and over and over again. So there are a number of examples where RNA tests are being employed through digital PCR.
Great, thanks, Varesh. There is a follow-up question here, actually, Varesh, that we'll put to you first. Can you comment a bit further as to the range of costs in dollar terms when quality of samples are qualified and ready to be used?
So the cost of digital PCR, certainly, I'll kind of go back to the time required to get a test running is going to depend on the complexity of the assay and the complexity of the markers that are being assessed. To put some perspective here, the cost of running a digital PCR reaction for a standard, let's say, two-target test or a two-target assay would be in the $10s to $20 per sample range for the materials. Just to give you context, there it is an order of magnitude less expensive than NGS typically. But again, the assay complexity will certainly determine the cost structure of running a digital PCR test.
Brilliant. Thank you, Varesh. Oh, apologies.
I was just going to say, and then certainly lab running costs are going to be on top of that, and that's something that maybe Biodesics could has more knowledge about. But all those costs together would aggregate into the running cost of that test.
Thanks, Fresh. Yeah, so just to add a perspective on the lab, I think for any test, whether it's NGS, ddPCR, or any clinical test, really what drives costs down in terms of the services is the throughput. So as we have, I would say, reasonably high volumes going through the lab, it's actually quite effective in the processes in the lab and how we manage the samples. So if you're running very, very few samples and not maximizing the platform use, whether it's ddPCR or NGS, the costs per sample are going to be higher. And that's just true for any assay.
Brilliant. Thank you, Gary. We have one last question here. Gary, I'll put this to yourself first. Have you done concordance studies to compare MSI between ddPCR and NGS testing?
So this is better for Dawn because the data Dawn showed in the table actually shows some concordance results. Dawn, do you want to add anything to that?
Hi, Gary. Yeah, thanks. The assays that I'm showing are very preliminary. It's very early days. We have not done a head-to-head with next-gen sequencing yet. It's certainly, definitely in our plans as things further develop.
Brilliant. Well, thank you all very much. That is all the time we have. A big thank you to Gary, to Varesh, and to Dawn. And a big thank you for everyone listening. We hope you've enjoyed the session. If you do have any follow-up questions or comments, please email myself at ben.carrington@hansonwade.com, or alternatively, you can contact Biodesics at the email link on your screen. A link to the recording of the presentation and the Q&A session will be made available, and you will be notified by email in the coming days, so watch out for that. Thank you all very much for your time, and I hope you enjoyed the rest of your day.